Hello, Ephs everywhere. My name is Henri Wheeler, and I am a Williams Class of 2003 alum and proud daughter of John Wheeler in the class of 1965. I currently serve on the Society of Alumni Executive Committee, and it is my pleasure to welcome you on behalf of Williams College Alumni Relations to the fifth and final installment in this lifelong learning series. These one-hour virtual lectures have been scheduled throughout the fall and feature Williams faculty and expert alumni from a variety of academic disciplines speaking on different topics. You can find the recordings of past talks and other virtual Williams offerings at alumni.williams.edu/virtual. A few reminders before we get started. If you have any questions during today's talk, please use the Q&A function at the bottom of your screen at any time. We'll have a dedicated time for Q&A at the end, but you can submit your questions throughout the talk. Please reserve the chat as a space to engage with the community and share any reflections or comments you may have. Remember to select all panelists and all attendees from the chat dropdown so that your message can be seen by all. And with that, I would like to introduce today's speaker, Professor Lee Park, the William R. Keenan Jr. Professor of Chemistry at Williams College. Dr. Park's lab research is in the area of inorganic and organic materials chemistry and nanotechnology, with a particular interest in self-assembling systems and device applications. One area of her work has involved the preparation of liquid crystalline compounds, both organic and inorganic, which self-assemble into electrically anisotropic structures. This approach enables Dr. Park to tune a number of specific characteristics of these one-dimensional materials, such as temperature range for self-assembly and the strength of intermolecular interactions by means of molecular design and synthesis. She holds a PhD in chemistry from MIT, and her research has been funded by NSF and ACS, the American Chemical Society. And if you understood everything that I just said, you are leagues ahead of me. I must confess that I took a total of one science class during my time in the Purple Valley. It was a science for the non-major class entitled Biology of the Tropics. But as a lifelong learner myself, I'm excited to continue my learning today from Dr. Park. We are so pleased to have her with us to speak about new materials via self-assembly, strategies for tackling cutting-edge problems based on things you learned in Chem 101. Thank you, Dr. Park.
So thank you for that introduction, and thank you to all of you who have taken the time to attend today. I see some familiar names in the chat. So I'm going to go ahead and share my screen and get myself set up. All right, so what I'm going to talk about today are some strategies for designing new materials. And in particular, I'm interested in materials at the nanoscale. And so I'm going to talk a little bit about what's interesting about working with materials at that size scale. And some strategies for making these materials. And you don't need to worry, because I don't think you really need to remember any chemistry that you might have in your background, although I did notice some chem major names coming up in the chat. All right, so we're going to talk a little bit about why nanoscale features and architectures are interesting. And then we're going to talk a little bit about some strategies for developing materials at this size scale. And then we're going to talk about some possible applications. Some of these are applications that are already in development. Some are pie in the sky at this point. I'm actually probably not going to have a lot of time to talk about the work that I do in my lab. So I'm going to be showing you a lot of applications that come out of other people's labs, but I hope that I'll have a little bit of time at the end to tell you a little bit about what I'm interested in. I hope that you will go ahead and submit questions to the Q&A, and I'll try to answer what I can at the end. All right, so this is just a scale bar to get us oriented in terms of the size scale that we're interested in. So up here is a meter, and you probably have a sense of roughly how big a meter is. It's approximately about a yard. And 1,000 times smaller than a meter is a millimeter. So that's typically the smallest gradation on a ruler that you might encounter. 1,000 times smaller than that is a micron. And 1,000 times smaller than that is a nanometer. And smaller than that is an angstrom. So I've put in an angstrom here, which is 10 times smaller than a nanometer. And I've put that measurement in because that's one that's sort of near and dear to chemists, because an angstrom is roughly the size of an atom. So if we're interested in building things that are best measured in nanometers, that is, if we're building on the nanoscale, then our building blocks are going to be small molecules or atoms. And just to give you a sense that, these are objects that are far smaller than we can see, right? So the limit of what we can see with the naked eye is in the micron range. And smaller than that, we need to use a light microscope. And this could just be the kind of microscope you might have used in an intro bio class. But the kind of structures that we're looking at are far too small to be seen with a light microscope. And so we have to use electron microscopy or atomic force microscopy to look at these features. So if we're trying to build things on the nanoscale, so those are very, very tiny features. So one of the challenges is how to do that. But one of the particular challenges is how to do that over very large areas. So that is, I'm interested in structures where the features might be nanoscale, but I would like to build them over very large areas. That is, I'd like to build them over the microscale or the macro scale. So just to take an example from work that I've done in the past, some of the architecture that you need if you're interested in looking at solar cells, for instance, and improving the efficiency of solar cells, is you need an internal architecture where you have features that are best measured in nanometers. But of course, you'd like to build enough solar cells to cover your roof and everyone else's roof as well. So you want to build these very tiny features over very large areas. And so that's really one of the challenges. Okay, so why is it interesting to look at things on this kind of size scale? So I'm going to give you just a couple of examples. Some of these you may have heard of before. So this is a picture of a gecko, and geckos are known to be able to climb up walls and climb on the ceiling. And the thing that's really remarkable is that you can have a gecko suspended on the ceiling, but that gecko can just pick up its foot and keep walking. And if you look at the wall or the ceiling after the gecko has walked by, what you'll find is there's no trace of anything sticky on the wall. That is, the geckos are not secreting some sort of a sticky substance out the pads of their feet. So there's nothing that we would recognize as an adhesive or as a glue. So scientists, of course, were interested in trying to understand how this could be. How does the gecko manage to do this? And if we zoom in using one of our fancy microscopes and look at the pad of a gecko foot, what you see is this. And you see that the surface is covered with these small hairs. And these hairs are on the order of microns in diameter. So these are smaller than you can see. But I think what you can see from this image is that those hairs actually split into smaller hairs. And at the very end, they actually split into these flat projections where these little flat areas are actually best measured in nanometers. So you have these little spatulate projections that are incredibly small. And it turns out it's this architecture of nanoscale features that gives rise to this adhesive property for the gecko foot. So there's nothing that we would recognize as a glue, but when the gecko puts its foot in contact with the wall or the ceiling, there's an enormous amount of surface area that comes in contact with that wall. And it's that surface area that leads to these adhesive properties. So of course, the next thing that scientists want to do is try to replicate this. And I'll tell you that we have figured out how to do that. And so there are now companies, in fact, that have figured out how to manufacture a surface that looks rather like the gecko foot. And they've established some proof of principle that you can actually use this to climb up the side of a building. I will say that this particular application is probably not the most important application that this kind of material will have. There are lots of other situations that we can imagine where having an adhesive that doesn't actually require a glue might be really interesting. And so there are a lot of applications, for instance, in biomedicine and tissue repair and other kinds of places that you can imagine making use of this kind of a material once you've understood how it works and how to fabricate it. So another example that I will give you is shown here. So these are lotus leaves, and Lotus leaves are known to be what's known as superhydrophobic. And what that means is that water drops, when they hit the surface of a lotus leaf, they beat up and in fact sort of tend to skitter right off the surface. And in trying to understand why this comes about, if we look closely at the surface, again, using one of these electron microscopes, we find that the surface of the lotus leaf actually has a structure that is not unlike that of the gecko foot. That is, you have a very detailed architecture that consists of these small bumps of certain sizes that are packed in a certain way. And so it's really this nanostructure that gives rise to this property of superhydrophobicity. In terms of applications of superhydrophobicity, if you have a material that sheds water in this way, that's a material that has self-cleaning properties. And so there are lots of applications that you can imagine for being able to put a self-cleaning surface onto a device or an object. So those are just a couple of examples to give you a sense of why this nanoscale architecture can lead to some really interesting kinds of properties. So then the next question is, how do we actually try to replicate those kinds of architectures? So we're going to start by looking at, how we make, a structurally interesting object at all. So here's an object that is created by simply starting with a block of material and removing all the parts that you don't want. So as a strategy, that's pretty straightforward. Obviously, this takes a good amount of work and effort. And you can see that you can come up with an object that has an awful lot of fine structure and a lot of detail. One downside for this object, at least in terms of the kinds of applications that I'm interested in, is that this is not easy to replicate. Obviously, if you're interested in this sculpture, then replication is not your primary concern. But for the kinds of applications that I want to talk about, replication is a concern. Okay, so this is one way to create an intricate object. So here's another example. This is a wood block print. And in this case, you start with a piece of wood and you carve away the parts of the pattern that you don't want. And so you can see that's something that these two methods have in common, that you're starting with a block of material and you're carving away the parts that you don't want. So this approach is known as top-down. So in the case of the wood block print, once you've carved your pattern, then you can ink your surface, and the ink is going to go on the parts of the surface that are raised. And you would selectively ink your surface, then you put your piece of paper down and transfer that pattern. Then you might clean off your block and ink some other parts of the surface. Now you're going to use the blue ink, and then you put your piece of paper down again and transfer your pattern. So the actual print that you've made is actually, if you got down on the side and looked at it, would actually be a multi-layer structure composed of these different layers of inks, but it was created by starting with that large block of wood. So this is still a top-down structure, albeit one that is easier to replicate, I think you'll agree, than this one. So the last example I want to show you is this. This is a lithograph. And in this case, you start with a piece of stone or maybe metal, and you put a thin coat of wax on the surface, and then you carve your pattern out of the wax. So again, it's a top-down method. But now when you've carved your pattern out of the wax, now you can flood the surface with ink. And the wax has the property of being hydrophobic, and the ink has the property of being hydrophilic. So the ink tends to be repelled by the wax, and so the ink will tend to flow into the regions where you've removed the wax. So this is a situation where you're still using a top-down method, but now you're using the chemical properties of the ink and the wax to control where the ink actually goes. So you can see, again, all three of these methods are top-down in approach, but we are improving things in terms of our ability to replicate the patterns that we've created. Okay, so these top-down methods are really powerful, and we can make a lot of very intricate structures using top-down methods. And this is, in fact, how computer chips have been made for many years. This process by which we make computer chips is not exactly the same as what I just showed you, but it's in fact very, very similar. And it's known by a technique that's known as photolithography. And we can make very fine features. We can make them very reproducibly. And we can make some really intricate kinds of structures. But we do run into some limitations using these top-down methods. And in particular, we would like to be able to make features that are even smaller. So as you know, we're always trying to make computer chips where the circuitry is smaller and smaller so we can pack more computational power onto a chip. And so we have reached the limits of what we can do with these top-down technologies in terms of this size scale. Another limitation of these top-down methods is that we would also like to be able to use different kinds of materials. These top-down methods tend to be best for hard materials, and we would actually like to be able to design nanoscale structures using soft materials. So those are a couple of limitations. The top-down methods are still very important. We still use them all the time, but we would like to figure out a way to complement them. So what we've talked about so far are top-down methods. So the complement to that is bottom-up. And in terms of bottom-up methods, this is meant to be just as, sort of an example of what that might look like. That is, you're going to start with building blocks. And your building blocks are going to be objects that, have the shape that you want. And then you have to figure out how to get your building blocks organized into the structures that you want. Now you can imagine if these are your building blocks, then it's going to take a fair amount of effort to get those built up into the larger structures that you're interested in. Now I told you that if we're interested in structures on the nanoscale, then our building blocks are going to have to be atoms and molecules. So we do in fact have techniques that allow us to pick up single atoms and molecules and manipulate them. But those methods are not going to be useful if you're trying to build a lot of something, right? So we can do it, but we can't, it's not a production method. So we need to think about different ways to get atoms or molecules organized into the structures that we want. So we take another lesson from nature. So these are some photographs of snowflakes. And what you need to understand is snowflakes, of course, are just composed of water, and water is a fairly simple molecule. But these snowflakes, these structures have assembled themselves. That is based on the structure of the water molecule and the particular properties associated with that structure. These water molecules organize themselves, they self-assemble into these incredibly elaborate structures, right? And the estimate is that there's something like 10 to the 19th water molecules in any one of these snowflakes. And in fact, these structures can be replicated. So in spite of what you've heard about snowflakes being unique, that's not quite true. And under the right conditions, you can actually replicate these structures. So this kind of self-assembly, the thinking about how to design molecules such that you can build in the information that will cause those molecules to organize themselves into the structures you want, that's really the strategy that we're going to take. Okay, so what kinds of properties are we going to try to manipulate? So we're going to keep it simple. And we're going to be looking at hydrophilic and hydrophobic properties. So water is a polar molecule. It's hydrophilic. It likes water. Something greasy, something like oil, is non-polar. It's hydrophobic. It doesn't like water. And so this is just a beaker with some water that has some food coloring, just so you can see it, with some vegetable oil. And you can see that these materials don't like to mix. So if they can, they're going to stay apart from each other. So we take advantage of this property already all the time in applications that you're very familiar with. So when we wax a car or you Scotch guard your winter boots and make it waterproof, right? What you're doing is applying a thin layer of something hydrophobic to the surface of that material. And because of that, the water droplets, when they land on that surface, they don't want to interact with the surface. So they tend to beat up and run off. And so that protects that surface. When you wax your skis, you're doing the same thing. You're putting a coating of hydrophobic material that allows your skis to glide over the snow. And high performance swimwear is also a material that is taking advantage of hydrophobic properties to allow the swimmer to glide through the water. It would make no difference in my swimming ability, but I'm told that for competitive swimmers, it makes a difference. So those are applications of just taking advantage of the fact that hydrophobic and hydrophilic materials don't want to interact. Okay, so we're going to now try to get a little more clever about that. So we can design molecules, or there are lots of examples of these molecules in nature, molecules that are what we call amphiphilic. That is, they have both a polar group as well as a non-polar group in the same molecule. Okay, so you can prepare a molecule that's amphiphilic. And then what you can do is put that molecule in solution, and then we can dip a slide into that solution. And the slide is coated with a thin layer of gold. So you dip that slide into that solution for maybe 30 seconds or a minute, and you pull it out. And what you have is your glass slide with its thin gold coating. The thing that happens is that the polar head groups interact selectively with the gold surface. And what you end up with is a gold surface where you have an extremely well-ordered arrangement of these molecules with the polar head groups bound to the surface and these greasy tail groups sticking up in the air. And the thing that's remarkable about this is you take your gold slide, which might be a few inches in area, square inches in area, and you dip this in your solution for a very short period of time. And you can organize these molecules based on this property of self-assembly very quickly, right? And so, this would be the equivalent of, taking yardsticks and managing to get them all lined up on end over an area of square miles. So it's really actually pretty remarkable. But here's the thing, what you've put on the surface is a molecular monolayer, right? This layer is exactly one molecule thick. And so when you pull your slide out of the liquid, you can't tell that you've done anything. You can't see that. So how do you convince yourself that something's actually happened? So what we can do is we can take a surface and we can put a water drop on that surface. And if you have a surface that is hydrophilic, then as I talked to you about on the previous slide, the water drop is going to spread out. The water drop is going to be happy to interact with that surface, and it's going to spread out, and it might have a profile that looks like this. But if you take a surface that's hydrophobic, as this surface would be with all these greasy tails sticking up, then the water drop, when it interacts with this surface, it's not interested in interacting with that surface. So the water will tend to bead up so that the water molecules can interact with each other as much as possible and minimize the interactions with the surface. So you dip your slide in this liquid. You can't tell that you've done anything by eye, but you can tell by doing this simple test, and you can see that you've changed the surface chemistry. Okay, so this is a very early image from one of the groups that originally was doing this kind of work. And in this case, they drew lines of the hydrophobic material along their surface, and then they put down water. So this is water with some fluorescent dye in it to make the picture a little more interesting. And you can see that you can control where the water wets the surface, right? And so in this case, you're able to make these square water droplets. And so that makes for a really fascinating image. But of course, square water droplets is not really, the goal. So what can we do with this? So what we might want to try to do is think about how can we pattern a surface? So we're trying to chemically pattern a surface. And we have a couple of options for doing that. So one option is that we can do what's called microcontact printing. So this is the same thing as taking a rubber stamp and inking it and printing that onto a piece of paper. The differences are that the ink that you're using is not chosen for its color, but for its molecular properties. And so you're inking your stamp with a molecular ink that's one of these amphiphilic molecules, and you are stamping it onto a surface so that you can chemically pattern a surface so that you'll have regions that are hydrophilic, regions that are hydrophobic. The other difference is that the stamps that you might use have features that are, again, much smaller than you can see. So on the order of patterns that might be best measured in microns. So that's one way to chemically pattern a surface. Another possibility is a technique known as dip pen nanolithography. And in this technique, you take a very fine tip that is atomically precise at the end, and you can dip it into one of these molecular inks, and you can use it just the way you would use a quill pen. That is, the ink will flow off the tip and onto a surface. And again, your ink is one of these amphiphilic molecules. So as it flows off the surface, it adheres to the surface in a very regular way. And so using this method, you can actually write patterns onto a surface that are one molecule thick and where your lines might be a few nanometers in width. So these are two methods that you can use for chemically patterning a surface with very fine features using this kind of a molecular ink. So if I have time at the end, I'll tell you about an example where I've used something like this. But I'm going to step back to show you another example. We can take these same kinds of molecular inks and we can load them into an inkjet printer. And just as if you have an inkjet printer, you can print out a gradient. So I've illustrated this here with a gradient from blue to purple. You can load your inkjet printer and print out a hydrophobic to hydrophilic gradient. And if you do that, then you have a very smooth transition between, a very hydrophobic end of your piece of paper or your substrate and a very hydrophilic end. So now if you imagine putting a drop of water onto this surface, then you can imagine that the left edge of this water drop is experiencing a relatively hydrophobic environment, and it's going to try to retract from that environment. But the right edge of this water drop is going to be experiencing a more hydrophilic environment, and it's going to try to spread out, try to wet that side. So as a result, this water drop is in fact going to move across the surface. So you can get this water drop to walk across the surface based on the chemical patterning of the surface. So you can do various things with this. You can get water to walk uphill by just tilting your paper up at an angle. And so this, and along with some other chemistry, is the basis of a whole range of things that are happening now known as microfluidics. So here you can see a short video clip of, this is just water with a dye in it, and you can see it traveling uphill into this reservoir. Here's another clip where the water is being separated into these channels and flowing out into these wells, okay? So what is this good for? So you may have heard people talk about labs on a chip. So these are microfluidic devices that are being explored and developed for doing biological assays, right? And the feature is that you can take very, very small amounts of a biological sample, whether that's blood or serum or saliva, for instance, and you can create these devices where you can control the flow of these fluids through these very small channels. And then you could imagine having different reagents in different reservoirs that might react with something in that sample and provide you a quick test of whether or not there's some disease marker in that sample, for instance. And the idea is that these assays could be done on very small amounts of sample. They could be done very quickly. They could be done away from a full lab. So there are a lot of potential advantages in developing these, and there are a lot of things that are already happening with these kinds of devices. So that's just one example. Another example, so this is fairly recent, and I thought I would play this clip for you. These are surfaces that have been coated with, again, hydrophilic materials. And these are water droplets that are splashing down on the surface. And so the really water droplets wet the surface and control the water droplets that are coming. And this has a lot to do with and as well as for converting into as an example where water drops and actually convert that motion to beautiful examples. All right. Figure out how to, there we go. All right, so here's just one more example that I came across relatively recently. There are parts of the world where there is not enough rainfall to support the population living there. And so scientists have been working on ways to harvest water out of the air. And so there are devices known as fog harvesters, and they are really, in a sense, just mesh screens that are designed to sort of trap the water that's in the fog. But what you need to do that effectively is a combination of hydrophilic and hydrophobic properties in this mesh. So you need it to be hydrophilic so that the water droplets and will condense well on this mesh. But you also need it to be hydrophobic because you need those water droplets to aggregate and actually run off. So people have been playing with these for quite a long time, learning how to develop them. And one of the things that they've more recently realized is that you actually need to incorporate hydrophobic elements as well in order to actually cause the water to run off so that you can actually collect it. So those are just some examples where we can make use of hydrophilic properties, hydrophobic properties to do some interesting things to address some real world problems and come up with some very cool applications. So now I want to move to thinking about how you actually create structures that are three-dimensional. So far we've been looking at patterning surfaces where you have a region that's hydrophilic and a region that's hydrophobic. So now I want to look at how you can take advantage of these same properties to create more intricate structures. So here's my beaker of water and oil that doesn't want to mix, but I can add an emulsifying agent, which is actually just an amphiphile, just the same kind of molecule that we've been talking about. And once I add an emulsifying agent, I can actually stabilize that mixture. All right, and you see this all the time. There are lots of emulsions that you're familiar with, milk and ice cream and mayonnaise. These are all materials in which you have small particles of the oily substance dispersed in the aqueous environment, right? And the emulsifier or the amphiphilic material is the thing that helps you do that. So how does this work? So this again is an amphiphile, and again there's a polar part and a non-polar part. If I take that amphiphile and I throw that into a beaker of water, the non-polar parts are not going to be at all happy. The polar parts may be very happy. And what they will do is they will self-assemble. The molecules will self-assemble such that the non-polar greasy parts can hang out together and ignore the water, whereas the polar groups can hang out with the water. And so what you form here is a structure known as a bilayer membrane or a bilayer sheet. And depending on the particular amphiphiles that you use and the particular concentration in solution, you can make different kinds of structures. So this kind of bilayer sheet is the kind of thing I was talking about here. So this is exactly what cell membranes look like in the body. You can also get micelles, which have a greasy cavity on the middle, on the inside, and you can get liposomes. So you can get different kinds of structures just based on this kind of combination of properties. And so again, these molecules might be quite small. They might be only a nanometer or two in length. And yet you can get this kind of self-assembly extending over into macroscopic size regimes. So there are cells that are big enough for us to see. And certainly in lab, we can make bilayer membranes over very, very large areas. So this idea of self-assembly, getting the molecules to organize themselves, is really powerful for the kinds of things that we would like to make. And examples of amphiphiles. So, you know, if you read the ingredients on your food packaging, lecithin and carrageenan and xanthan gum, these are words that you probably recognize. And these are all emulsifiers. They're in your food products in an effort to stabilize the emulsions so that your ice doesn't separate into ice crystals and sort of fat globules. I just want to mention soap. So when we think about soap, we're trying to clean, you know, if you're trying to clean a greasy pan, you're trying to wash away something greasy with water. So that's not going to work very well. So you add soap, and what happens is that the soap forms these kinds of micelles, and the greasy bits get encapsulated in the greasy interior of these micelles. But then the outside of the micelle is very polar, and so that gets washed away with water. All right, so that's how soap works. Okay, so what I'm talking about so far are small molecule amphiphiles. We can do the same thing with larger molecules, okay? So we can make what are known as block copolymers. And so if you take two different polymers, and you don't need to worry about what these are, one of these is polystyrene, the other is polymethyl methacrylate, but the important thing is that they are different and they don't like each other. But you can make a block copolymer, so a very long molecule where you have one part that's all made up of the polystyrene and the other part that's all made up of the other polymer, and you can stitch them together. So when you make this kind of material, this is an amphiphile. It's a much longer amphiphile than the ones that I've been talking about, but these amphiphiles will also do their best to organize themselves in a way to maximize the favorable interactions and minimize the unfavorable interactions. So if you take one of these block copolymers, you can actually control, based on the length, the relative length of the two blocks, you can control exactly what kinds of structures you get. So depending on the relative amounts of the blue versus the red polymer, you can get alternating sheets. You can get columns of one material in a matrix of the other. You can get spheres of one material in a matrix of the other. And just as I showed you for the small molecule amphiphiles, you can get bilayer sheets, or you can get micelles or liposomes. With these large amphiphiles, you can get very similar behavior, and you can control that. So you can control what kind of structure you get. Okay, so now I can talk a little bit about some applications of this kind of chemistry. So this is one from a colleague of mine in my department, Sarah Go. So she makes amphiphiles, and in her case, the amphiphiles she's making actually happen to have 3 parts to them. And so there are 3 parts to them, but these behave just as I've described. If you throw this material into water, it will organize itself such that the hydrophobic parts will be encapsulated on the inside, and the hydrophilic parts will be on the outside. So the reason that she's interested in this is that a lot of the drugs that we take, the medicines that we take, tend to be somewhat hydrophobic in nature. And you're trying to get them into the human body, which is a very hydrophilic environment. So there's often a little bit of a mismatch, and that's part of the challenge in thinking about how to administer drugs. And it also has implications for how much of a drug you need to take. So what she's doing is looking at designing these kinds of amphiphiles. And then when she throws these into solution with a drug molecule, then she will form these kinds of micelles where the drug material will be encapsulated, will be trapped in this sort of more greasy interior. So it works exactly the same way the soap micelles do. And then the idea is that these micelles, which are now very polar, can be transported into the body. And so this is a TEM image, a transmission electron microscope image of one of the micelles. And you can see the cavities where the drugs might be encapsulated. And then, of course, she designs these in such a way that to think about then, once these are in the body, how do they degrade such that the drugs are released? So that's an application of, again, all of these ideas of self-assembly and thinking about what you can do with them. So going back to this image, so we can control the morphology, that is the particular architecture that we get by controlling the length of these two parts of the molecule. This image is an atomic force micrograph image of the surface of a film of one of these polymers. So this is a polymer film made of one of these dye blocks. And this is actually a structure that looks like this. It's these columns, all right? And you can sort of get a sense of that. What you can understand is that you're getting these very regular features, right? You're getting these very regular stripes. They're very uniform in size. The individual stripes, the light and the dark, are the red versus the blue polymers, all right? So you're getting this very interesting organization. It's kind of beautiful. The thickness of these stripes is on the order of tens and hundreds of nanometers, so very small features. So you can take this kind of system and you can now play more games with this. That is, you can design your polymers such that you can take advantage of other properties that these materials might have. So for instance, they might be different in terms of solubility. So here is an image where somebody has created a film that looks like this and then dissolved away one of the polymers and left behind the other. And so you can see that what you're now creating is an architecture where, so this scale bar here is 2 microns, right? So this whole thing might be, let's say 10 or 15 microns. So these walls are, maybe tens of nanometers in height, okay? And so you're creating this nanoscale architecture where you've now built this network of walls. So that's pretty amazing. Now you might look at this structure and you'd say, well, there is some regularity to this structure, but perhaps for some of the kinds of things that you want, this isn't regular enough. So this is where we can bring in these ideas of the surface patterning that I was talking about before. That is, you can take a surface and you can pattern the surface, chemically pattern the surface using some of the methods that I described before. And here's what you might get. So this is a surface where this part on the right was patterned in some way and the part on the left wasn't. And then a polymer film of the type on the previous slide is cast onto that and allowed to organize itself. And what you can see is clearly on the pattern side, the polymers have organized themselves into very regular stripes. And on the other side that hasn't been patterned, you get the pattern that I was showing you before. And the remarkable thing about this is the chemical patterning, remember, might only be a single molecule high. And yet your polymer film that you've put on top of it may be microns thick, right? Might be thousands of times thicker. And yet that self-assembly process exerts its influence all the way through that polymer film. And that's really quite remarkable. We can play other kinds of games where we can learn to put down different kinds of patterns to create very regular structures. So here we've gotten these polymer films. I should not say we, this is not my work, but scientists have gotten these polymer films to turn sharp corners, for instance, or to create extremely regular structures. And you can see, the kinds of features that you're building, this scale bar is 50 nanometers. So what people are learning how to do is to create these very fine structures, but over very large areas and through a very great thickness. And so that's really kind of a remarkable set of things to be able to bring together in one process. So with that, I can tell you just a little bit about how this relates to work that I've done. And so I have done some work in the area of organic solar cells. And in some of that work, I used some of the techniques that I've already told you about. So in this slide, this is an illustration of chemically patterning a surface, which I did with different kinds of size patterns, both through the microcontact printing, which made sort of micron size patterns, as well as through the dip pen nanolithography, which made nanometer size patterns. And then you can coat that polymer film, coat that surface with a polymer film, and you can let that polymer film anneal and look at the self-assembly that you get. And again, the goal of this project was really to try to impact the interior architecture in that polymer film in order to improve the efficiency of a solar cell. So that was one kind of work that I did some years ago. What I've been doing more recently is related in that I'm looking now at gold nanorods. So these are individual gold nanoparticles that are rod shaped. They're about 50 nanometers in length and maybe 10 nanometers in width. And what I'm working on is looking at chemically functionalizing the surface of these nanorods in order to get them to self-assemble. So I'm trying to think about how to chemically alter the surface so that I can get these nanorods to line up and aggregate in certain ways, and possibly get them to aggregate into structures that will be useful to me. And in particular, the thing that I'm interested in is that gold nanoparticles and gold nanorods have very interesting optical properties. And by getting them to aggregate in very controlled ways, you can really control the absorption profiles of this material. And in controlling these absorption profiles and the structures that you get, you can conceivably combine this with some of this solar cell work to actually think about how to better create a more efficient kind of solar cell device, one that can absorb light very efficiently, and then harness that energy in the light to make an efficient solar cell. So that, in a nutshell, is some of what I've been working on. And with that, I think what I'll do is just leave you with this slide. So this is an image that I made using one of those dip pen nanolithography techniques. So this is an image of buildings that I hope that you will recognize. It's written in one of those molecular inks. So it's exactly one molecule high off the surface. And it's on a piece of gold. And this whole image is about 10 or 15 microns across. So this is not something you can see. And the colors are appropriate, but false color that I included in the processing after the experiment was done. Okay, so I think I will stop there, and I will go ahead and see if there are any questions in the Q&A. I got to figure out how to do that. Let's see. I think I need to change this. All right, so let's see. What kind of questions do I have here? So, okay, so one question is, do we have an atomic force microscope or an electron microscope? Yes, we do. So we are very well equipped here. We have fairly new transmission electron microscope, as well as a scanning electron microscope, and we have a couple of atomic force microscopes as well. So a fair amount of this work is work that we can actually do here. So let's see. What else? Let's see. So there's a question about dust. So yes, if we're working on materials at this size scale, then clearly any amount of dust is going to be a huge obstacle. So yes, so when you're working with things on this size scale, that can be a real problem. And so some of the techniques that I described are really, so for instance, the dip pen nanolithography as a tool, that is probably not something that's ever going to be kind of a production kind of approach. So that's really something you would use when you are exploring, the possibilities for doing this kind of surface patterning and looking at the impacts on self-assembly. But if you were really, in the business of, if you were really actually ready to build a solar cell, that is not going to be a technique that you're going to use. Partly, just the time frame, that's much too slow a technique. And as you say, the kinds of normal things that you might find, a little bit of dust is going to be a big problem. So really, the goal in all of this is also to find techniques and processes that are more robust than that. That said, I will say that the nanolithography methods that we use to prepare computer chips, for instance, we can build incredibly fine structures, on a very large scale. And chip manufacture facilities are, of course, very specialized facilities, and you operate under positive pressure. You're trying to keep out all the dust. So there are various approaches to thinking about that, but it's true that those would be real problems. Let's see. Okay, a couple of these get a little bit more involved. So let's see. Okay, so there's a question about how to do some of this in the high school environment. Some of this you can absolutely do in the high school environment. So for instance, the surface patterning, making the self-assembled monolayers, that's actually something we do in our intro labs, and I've taken on the road to different high schools and middle school environments. So that's actually quite easy. If you want to write to me later, I'd be happy to tell you how we do that. There are some other kinds of things you can do to illustrate the ideas of chemically patterning surfaces. And actually, if I share my screen with you, I might be able to show you. There was one thing I was going to show you, and I decided that we didn't have time, but maybe I can show you one last slide. Let's see. So, okay, so this is actually just a petri dish, a polystyrene petri dish, right? And it's covered with a foam sticky, and I treat it with a Tesla coil. So we're chemically altering the surface. And if I can get this little video to play, after you treat this with a Tesla coil, I think what you can see is that the water will selectively flood certain areas and avoid other areas. So this again is, sort of a very simple kind of example of this kind of surface patterning that you can do, and that you can do, kind of at the middle school or high school level. And again, I'd be happy to give you some more details about that. Let's see, what else? So there are a number of questions that ask about details in terms of some of these microfluidic devices. So that's a big area, and I will have to say that I am not expert in what's going on in those areas. I know that there are companies developing different microfluidic devices to test for all kinds of things. And so some of the specific questions that people are asking about particular kinds of tests, my guess is that there are people working on a lot of them, but I'm afraid I really don't know. And many of these microfluidic devices are in fact, I think just starting to be used. So I think there are some really promising possibilities out there. What else? So there are, let's see. There are various questions asking me if I'm ready to patent anything. I'm afraid that I'm not. So the work that I'm doing is really, really on the fundamental end. I'm really trying to look at ways to think about the self-assembly properties and how to impact the self-assembly properties at the moment in gold nanoparticles. There are certainly potential kinds of applications for that if we are successful, but there would also be an awful lot of things to work out, before you actually got all the way to a better solar cell. So I'm probably not set up to take it all the way. So that probably wouldn't be coming out of my lab. Let's see, what else? And then there are a couple of people talking about, other things that they've read about. I see some names that I recognize who may know more about some specific details about what the latest is on some of these drug delivery systems. But I think that there's some really interesting work there. Okay, what else? So in terms of students in my lab, usually I have a number of students in my lab, both in the summers and during the academic year. This year, like all years, is a little bit unusual. So in my lab at the moment, I actually don't have any students because I was actually supposed to be on sabbatical this year. So I hadn't taken any students. But I'm hoping that, hopefully the world will get a little bit more back to normal and we'll be able to take more students into our lab, maybe starting this summer, but hopefully next year. So yeah, I'd love to be able to get back to work with students. So the self-assembly of snowflakes. So water is a molecule. It's a very small molecule, H2O, only composed of 3 atoms. But the water molecule is bent. It's bent in shape, okay? And it's bent and it's an oxygen bound to 2 hydrogens. And because of the electronegativities of those different atoms, the oxygen end of the molecule is a little bit negatively charged and the hydrogen end is a little bit positively charged. So the molecule itself has this property of being polar. And because it has this property of being polar, the water molecules, when they start to crystallize, so when they're water, when they're liquid, they're all moving around. But when they start to crystallize and sort of get locked into fixed positions, the polarity of the molecule governs how that happens. And so what ends up happening is that the slightly negative end of the molecule tends to align with the slightly positive end of the next molecule. And so you get a certain organization happening there. And if you can crystallize your snowflake slowly enough to give all the water molecules enough time to sort of find a position that suits them, then you can, make these kinds of beautiful crystals. Let's see, what else? So why do these patterns happen? So again, you're taking advantage of these different properties of these different materials. I talked mostly about polar versus non-polar materials wanting to avoid each other. And when you get to these complicated systems like these block copolymers, you have this very long molecule. And so it isn't possible, because these very long molecules are very entangled. It's not possible for them to organize themselves in such a way that you completely get all of one kind of one part of the molecule over here and the other part of the molecule over here. The chains, because they are entangled, can't quite allow the molecules to get as far away from each other as they would like. So it's a combination of these different effects. It's that the different parts of the molecules are trying to avoid each other, but they are also entangled. And so it becomes, there are various things going on in terms of the intermolecular interactions or repulsions, and the kinds of structures that allow you to maximize the energetics that are favorable and minimize the energetics that are not favorable. And part of what's interesting about a lot of this is these very intricate patterns that you get that are reproducible. You actually see these patterns in other parts of the natural world. So these are not random patterns. They are very beautiful, but they are not random. And so that's also just an interesting aspect of this. How much has Chemistry 101 changed in the last 20 years? Well, for one thing, it's not actually called Chem 101 anymore. So I think that introductory chemistry here and elsewhere, in many respects has many similar elements. There are certain fundamental concepts that we still have to cover in terms of structure and bonding. How do atoms come together to form molecules, and what are fundamental properties that arise from different kinds of molecules? So there's still a lot of basics that we need to cover. But one of the tensions, I think, in any institution is of course, there's a lot of really interesting new chemistry that's going on in the world, and you would love to be able to communicate that to students and show them what's the most exciting thing. What are the most exciting things happening in the field? And so trying to figure out how to introduce new ideas, new research, even into an intro chem course, and trying to think about, in order to create the time to do that, what else might you have to let go of? I think that's a tension in a lot of disciplines, trying to figure out how to find that right balance. We do try to do new things. We're actually, in the process of different kinds of conversations in the department, trying to think about whether or not we want to make some big changes to intro chem, to how we run our labs, to the kinds of things we have students doing in lab, trying to think about laboratory experiences that will be more research-like, trying to show students, different kinds of applications of chemistry. So I think that's always, an ongoing conversation. So if you took intro chem, a long time ago, there would certainly still be things on my current exam that you could do, but there might also be some things that you didn't recognize. All right, so let's see. I think we have maybe time just for one more question. Okay, so somebody is commenting that I keep anthropomorphizing my atoms and molecules. You're quite right. I keep saying that the atoms and the molecules want to do this and want to do that, and that is perhaps not the right way to talk about it. But that said, the atoms and molecules are responding to forces that have to do with the energetics, have to do with kinetics, and so they are, trying to move to an energetically most stable configuration, and that is something that they are in fact working towards, not through any will, but nevertheless, there is a drive to move to an energetically more stable conformation. And we are always just trying to either understand what that conformation is or take advantage of it. So I think that that's about it in terms of time.
Thank you so much, Dr. Park. We really appreciate you taking time out of your busy schedule to share your work with the Ephs community. And just as a reminder to everyone, this lecture and others can be found at alumni.williams.edu/virtual. And we hope you'll join us for more webinars in the new year as the Society of Alumni turns 200. And we look ahead to a year of celebration, grappling, storytelling, and thought-provoking programs to mark our bicentennial. In the meantime, thank you again for joining us, and please take good care of yourselves.